Hi, thank you, Eugenia and Ventura, for the kind invitation. It's fantastic to be in this wonderful city, beautiful hospital, in this very nice audience. So I've been asked to try to reconcile the microbiome studies in HIV with the pathogenesis of aging, inflammation, in the context of HIV-infected patients. And it's going to be frustrating for me to summarize the evidence because there are plenty of beautiful studies. And it's always a good exercise in the microbiome presentations to step back and get rid of our classical training in infectious diseases linking microorganisms with diseases. And it's probably a good idea to imagine ourselves as ecosystems, because indeed we are the trillions of microbes that inhabit ourselves are so complex as any other ecosystem. And for example, here you have a grass close to Portugal, Cáceres, in Spain, and this grass is mainly formed by this leguminous, which is very good for shepherding, and it forms a very stable ecosystem, resilient to perturbations, and perennial. It doesn't change over the year. And if you look here, you see that there's a huge change. Okay, here, because here this grass was digged 3 years ago, and this grass changed into this other ecosystem formed by a multitude of flowers, which are opportunistic. They die every summer. They change. It's a very unstable ecosystem, and actually it's also very resilient because it's very hard to restore the ecosystem to the previous state. And these changes in floral ecosystems following perturbations can even be more dramatic. And here you have this plant. It's called viborera in Spanish. I don't know in English, sorry. So you see that it has a very slim stem with purple flowers. And this is what it happens after a single cycle of herbicides. These plants mutates, puts dozens of stems together, and becomes resistant to herbicides for generations. And this is like a frightening reminder of how resistance to antibiotic arise in clinical practice. And well, the floral ecosystems are very nice to think about ecology, but actually in the gut everything is dark, is humid, and there's a very strong competition among microorganisms for nutrients. And it's really hard and difficult to study how these microbes interact between them, and this is probably one thing we need to study more in the field, and some groups have already started to study the prey-predator relationships among bacteria. And last is even more important to understand what these microorganisms are actually doing, because this is what actually matters for clinical practice. I mean, are these bacteria leading to positive or harmful effects? And actually there's a very strong interplay between the microbiota and our body, and the immune system is one of the strongest interactions. I won't dissect this in detail, but the key message is that there are several components of the gut microbiota that are able to induce the Th17 response. This is like an accelerator in the immunity. It promotes the response against microbial invaders, and actually it's been seen that the microbiota is able to amplify this response and to amplify autoimmunity. But in contrast, other components of the microbiota, they do just the opposite thing. They are potent T-regulatory inductors, which is like an immunological brake. And among them, there are salient bacteria, such as the short-chain fatty acid producers, which digest the dietary fiber into these metabolites, which are able to induce the Treg response, so decrease autoimmunity and the Th17 response. Actually, these interactions are very robust, and it seems that they have very strong implications, and probably behind these immune interactions, there's the link between the microbiota and many diseases, including autoimmune diseases, cancer, metabolic diseases, atherosclerosis. And it's also true that the microbiota is sometimes oversold, but there are people who have even suggested that the bacteria can even influence how do we feel like and who do we like. So that's something to take in mind. So what are the changes throughout life in the microbiota? So we're born with a very highly diverse, we are born free of bacteria, but we are soon colonized by a multitude of bacteria, and there's a strong immunological and nutritional pressure to select some bacteria. And over life, our microbiome changes following physiological changes, following perturbations, antibiotic treatment, diseases, sedentarism, diet, and so forth. And at the end, the microbiota of an old individual shows a very high variability among individuals. The alpha and beta diversity is different compared to younger individuals, and in some there's decreased firmicutes, and among them decreased short-chain fatty acid production and increased bacilli, although the functional consequences have not been well addressed as far as I know. But it looks like there are different microbiomes in the elderly, and these microbiomes correlate with frailty, which is interesting from a clinical perspective. There's even a microbiota signature associated with frailty, predominance of Egertella and depletion of Faecalibacterium, which is one of the main short-chain fatty acids producers. These bacteria have also been linked with inflammation and also with neurocognitive dysfunction, even in studies in twins with and without dementia, which is really impressive to do as an study. And what's the link with HIV? Well, HIV, we all know well the story. The epicentrum of the inflammation is probably the gut. The gut is damaged very early after the infection, and this results into loss of these Th17 cells and loss of the enterocyte barrier function. And probably because of bacterial translocation, as Julia explained, we've got this activation of the innate immune response, which has been linked with comorbidities. So there are different components of the microbiota which have been shown to influence these pathways, but also it is reasonable to assume that if we have such a strong perturbation in the habitat of these bacteria, we will see changes in the composition and in the function of these bacteria, which may promote this vicious circle of inflammation and viral replication. So in the past years, there have been a lot of studies trying to elucidate which is the microbiome of HIV-infected individuals, and there are actually great methodological issues about controlling confounders. So the summary is that there are some bacteria that seems to be enriched consistently across studies in HIV-infected individuals. HIV-infected subjects have increased abundance of pathogenic bacteria, pathobionts, increased proteobacteria, and these are changes that we see in the elderly. And there are a lot of studies where we saw increased Prevotella, and this is like the rainbow bacteria. This is a great contribution from Ursicaisa, because they found that this bacteria is enriched in MSM and seems to be linked to sexual preferences. So it was a main confounder in our studies. And in contrast, there is depletion of lactobacillus and bacteroides, which is, and short-chain fatty acid producers, which is something we typically see also in the elderly. So there are some similarities between the HIV-infected dysbiosis and the gut microbiota changes in the elderly. So I'd like just to give you an overview of how and why this could be relevant for HIV care. So we all try to think about the gut when we hear about the microbiota, but there are actually a lot of, or some papers analyzing the microbiota in other compartments in which they've been shown that there's a very, there's influence of the microbiome in inflammation at this site. So something which is very intriguing is the blood microbiota. This hasn't been studied well because of methodological issues and contamination with PCR, but there's a paper in HIV-infected macaques showing that early after HIV infection, there's a shift in the bacterial products that they are translocated to the gut, and surprisingly, there's a peak of bacterial translocation that precedes 1 week the peak of viremia, suggesting that the bacterial translocation amplify inflammation and the HIV target cell recruitment. And actually, they were able to find some HIV controller macaques and to induce the loss of virological control by inducing bacterial translocation. So it is likely that bacterial translocation is promoting viral replication since very early stages of the disease. And what we've seen, this is unpublished, is that there is also a microbiota signature in the blood of HIV-infected individuals with predominance of proteobacteria, and there are biomarkers of immune recovery. When we look at these bacteria in naive patients and we follow them for a year, we see that enrichment for lactobacillus predict better immune recovery, and depletion of actinobacteria is also is a risk factor for a poorer immune recovery. And these bacteria correlate with inflammatory markers, so this is something that probably deserves to be studied further. The other compartment is the vagina. This is really interesting because we already knew that there's increased risk of HIV acquisition, not explained by sexual risk behavior in many parts in Africa. And Lindemann group found that this seems to be predicted by increased cervicovaginal inflammation. Many pro-inflammatory pathways were induced in the cervicovaginal fluid of the women who became infected later on. And Melissa Nattar was able to explain the cause of this inflammation. They found that the microbiota in women is mainly distributed in 4 cervicotypes, one with predominance of lactobacillus crispatus, another one with lactobacillus inner, and 2 of them with predominance of anaerobes, Gardnerella and Prevotella vivia. And basically, they found that when the main component of the vaginal microbiota is Prevotella, there's induction of a lot of pro-inflammatory pathways recruiting presumably HIV target cells. So this is very exciting because we are facing a modifiable risk factor for HIV acquisition. And the penis is a neglected site in the microbiome research, but there's been papers also looking at what happens after circumcision. We already knew that circumcision protects of HIV acquisition by men. So they were able to demonstrate that after circumcision, the anaerobes drop in the penis, and the proportion of anaerobes correlate with the risk of HIV acquisition with the concentrations of IL-8, which is a very interesting cytokine because any cell with toll-like receptors can produce this cytokine, and it mediates the recruitment of neutrophils. So again, we have bacteria, anaerobes, that are promoting inflammation and the recruitment of HIV target cells, and this is also a modifiable risk factor. So the lung is being less studied, but there are very interesting papers showing that the increased risk we see of bacterial infections and tuberculosis in HIV-infected patients may be explained by induction of regulatory T responses suppression, the specific immunity against tuberculosis, and this seems to be mediated by the production of butyrate by the anaerobes. It seems also that there are alterations in surfactant production mediated by impaired metabolism of the precursors, and there's also decreased cellular immunity linked to bacterial pneumonia, which has been explained by altered linolate metabolism. So in sum, we have some pathways which could explain something that is not explained today, which is why suppressed patients with good CD4 counts have increased risk of tuberculosis and bacterial pneumonia. And this could even, although this is speculative, why we see so dramatic COPD disease and increased risk of lung cancer not explained by smoking. So the butyrate leads us to the gut, which is the better studied compartment. So I just wanted to outline 3 pathways by which the microbiota can influence inflammation in HIV. One, as I mentioned before, is the short-chain fatty acid production. So these metabolites are produced in the gut by some bacteria, and among them, Fecalibacterium prosnitzi is a major butyrate producer. This is a very dominant commensal in the healthy gut, and it's able to digest the dietary fiber into this small molecule, the butyrate, which is very interesting because butyrate is the main source of energy for the colonocyte, is an anti-inflammatory molecule, and is also even able to inhibit the histone deacetylases. Martha talked about them before. And this is kind of a remote control of gut bacteria of our gene transcription activity. And we actually found that HIV-infected patients have decreased abundance of these bacteria, decreased levels of butyrate, at least in our study, and this correlates with the extent of histone acetylation in blood, suggesting that this is one mechanism at which the bacteria could be influencing inflammation and immune recovery during ART. The second pathway is the production of trimethylamine. This is a metabolite, also which is also derived from the diet, from phosphatidylcholine and carnitine. Some bacteria produce this metabolite, and in the liver is oxidized and is a very potent pathogenetic metabolite. And vegans actually don't produce this metabolite. So there was the hypothesis that maybe this dysbiosis in HIV could influence the increased risk of cardiovascular disease we see in our patients. And at least one study had pointed before that the plaque burden in HIV-infected patients is well correlated with the concentrations of these metabolites. And a few weeks ago, a paper from a German group focused on the microbiota of HIV-infected patients with and without cardiovascular disease, and they found up to 50 bacteria who were significantly different, more abundant or less abundant in the population of patients with cardiovascular disease. And the third important pathway is the the tryptophan catabolism, and this was one of the first papers published in this field in 2013. So this enzyme The indolamine deoxygenase is constitutively expressed in macrophages and dendritic cells in the mucosa, and it's able to catabolize the tryptophan into other catabolites which are immunologically active. They induce the Treg response, so they allow for microbial translocation, and actually the biomarker of this pathway, the kynurenine-tryptophan ratio, is a strong and independent predictor of mortality in HIV. So the San Francisco group was able to identify in HIV-infected patients up to 140 bacteria encoding the enzymatic machinery to reproduce this human pathway, and actually this correlated with inflammatory markers. And in a different study in elite controllers, it's been shown that this pathway is preserved in elite controllers. So it is probably very relevant for HIV immunopathogenesis, the effect that the microbiota can exert on this pathway. So the bad news is that most of studies have focused on the sequencing, 16S sequencing data, and this allows you to understand who is there, if you have Ewoks or imperial soldiers. But to know if these imperial soldiers are doing something bad, you actually need to go deeper into the functional studies, and you may want to study the transcriptome, the proteome, or the metabolome, so that you can deduce if the gremlin is quiet or you have fed him after midnight and it has become into a bad creature. So for this you need other kind of studies, and this was our first study in the field. We just looked at the metagenomic data using metagenomic shotgun to understand what genes were more represented in the HIV-infected populations. And I think the first thing I learned in this field is that this biosis is not necessarily a bad consequence in HIV, because we found that, okay, there was high abundance of some pathways belonging to inflammation, LPS synthesis, but there was also enrichment in the HIV-infected patients for antioxidant pathways, synthesis of cofactors, vitamins, suggesting that the microbiota is trying to repair a harm is going on. And this has been a very nice contribution from Irsi Kaisa group in a very beautiful paper in which they have also profiled all the genes in a very fair amount of patients. And I think their interpretation is something similar. They found that patients with very low nadir CD4 also have lower gene counts in their whole microbial communities, and this correlates also with an adaptation to oxidative stress and to inflammation. So we must be cautious about interpreting this biosis as a bad consequence of a disease, because actually it could be also an adaptation doing beneficial effects on the host. And this is a different approach. This is a proteomic study. When we compare different diseases and healthy controls, we found that in the fecal microbiota, 80% of bacteria in healthy subjects are dormant or died. And in contrast, in C. diff infection and in HIV, a large proportion of bacteria are activated. And when we did metabolomics, we found that some bacteria seems to be contributing to immune repair, to immune recovery by acting as anti-inflammatory buffers, accumulating within the bacteria pro-inflammatory mediators or anti-inflammatory mediators such as biliverdin or oleamide, while in subjects who are not responding well to ART, these bacteria are not acting as anti-inflammatory buffers. And I think this is very interesting since it gives you some insight on how the bacteria could be influencing immune restoration under ART. And this is hard to understand, even for those who are involved in the papers. But another approach is to try to integrate different omic techniques, and here we try to combine 16S data with transcriptomics and metabolomics to see which pathways depend on what bacteria. The findings, I think, are consistent with the previous message. We found that there are some bacteria which seem to be contributing to inflammation or expressing pathways related to resistance to oxidative stress, but there are also other bacteria which are doing just the opposite things. They are they are resistant to inflammation, they are anti-inflammatory, and they correlate with markers of T-cell activation. So a different approach is you take this bacteria and you go to the lab and see what happens when you do immunostimulation experiments. And this group from Colorado has been able to purify the bacteria from the stools of HIV-infected individuals, and they've seen that when you compare what happens in monocytes when you stimulate them with bacteria from HIV-infected individuals as compared with bacteria from HIV-uninfected, the activation of monocytes and T cells is much higher when with the microbiota from HIV-infected individuals, suggesting that this microbiota is actually pro-inflammatory. And they've also been able to see it doing fecal transplant with the microbiota from HIV-infected and uninfected MSM to mice, and they've seen consistent data showing that fecal transplant from HIV-infected individuals elicit higher T-cell activation than when we use the stools from HIV-infected individuals. So I believe that clinical trials are necessary to fully understand what's the feasibility of manipulating the microbiota and the clinical consequences. We probably have different endpoints related to lung diseases, to genital tract problems, cardiovascular disease, to GI tract, and to inflammation and HIV immunopathogenesis. And we may want to use prebiotics, probiotics, phages, antibiotics, fecal transplants, or to selectively use the bacterial metabolites that seem to be playing a role. So we've tried prebiotics, and it was surprising to see that when we looked at the effects of prebiotics 6 weeks in the same patients by 16S sequencing or by proteomics, we see a different picture. Using 16S sequencing, we detected some changes only in ART-naive individuals, but using proteomics, we found that in all groups, HIV-infected, uninfected, treated, and naive, there was a very strong activation of bifidobacteria, although we were unable to see changes in inflammatory biomarkers after this 6-week intervention. So we decided to do a larger trial, combining in a high-dose cocktail of prebiotics, probiotics, oligonutrients, omega-3 and 6 fatty acids and essential amino acids, which were given for 1 year to late presenters, since we believe that this was a population that may benefit from from boosting the immune recovery. And it was great to see that ART is really effective, all the biomarkers improved, but it was a bit disappointing to see that we didn't achieve anything in terms of the probiotic cocktail compared to the placebo. There was no signal of an effect of markers of inflammation, bacterial translocation, T-cell activation, and we didn't see even changes or only very small changes at the gut microbiota using 16S sequencing. So we were a bit frustrated, but there's probably another way to try to deal with microbiota manipulation. And Somsukma and Ivan Bukovic in San Francisco did a small trial with 6 patients using fecal transplant by a colonoscopy. And it was also a bit disappointing to see that the extent of engraftment is very limited compared to what you see when you give FMT to patients with C. diff diarrhea. This means that you achieve fecal engraftment from the donor, and this means that the extent of engraftment was very limited. So we are facing probably different problems. C. diff infection is like an arid field. There's no competitors. There are only some weeds growing up. So it's probably easier to repopulate this field than if we go to HIV or to chronic diseases. This is the Monegros Desert, very close to here. And here, what you have is an ecosystem which is adapted to a arid and continental and extreme climate. And you can't pretend to plant here a tropical forest because simply the conditions are not the conditions you need. So we are going to need to be very creative for interventions. So we designed this trial. This is fecal transplants by oral capsules from 3 selected donors based on their microbiota, and we are trying to reseed the field little by little to introduce changes sequentially, which is something that ecologists always say that it's very important to change any ecosystem. And we are just now analyzing these results. The trial has been completed. So as take-home messages, we probably need to think more about the microbiota as an ecosystem. It is clear that it's a very strong immune modulator. There are similarities between the microbiota and HIV and the elderly. It's been linked with T-cell recovery and inflammation during ART. It influences by promoting inflammation, HIV transmission in men and women. It affects the risk of lung diseases. But we need to understand better the functional implications. We need to assess more interventions, and it's either way a really exciting area of research. I'd just like to thank all my collaborators in different parts who are contributing to these studies, and thank you for your attention.